Welcome everyone. Assalamu alaikum wa rahmatullah. I would like first to express my sincere words of thanks for MEMS for their substantial invitation to deliver this webinar. And I would like also to extend my appreciation for their effort in promoting these meetings between Arab and other Middle East countries. Today we are gathering participants from 12 different countries all around. 46 participation should be online. I am Dr. Sami EL KHATIB, Assistant Professor in Biotechnology and Assistant Dean of the School of Art and Sciences at the Lebanese International University, located in the provincial region of Bekaa, with large spaces, welcoming people, and stunning weather all over the year. I'll be talking about the evolution of genome editing technologies, and will be focusing on the CRISPR-Cas technology, nowadays considered as an effective and very functional tool for genetic manipulation and biological research. Our meeting will be directed toward four major axes. The first one is an overview about the concept of genome editing, the major tools currently used in the field, and will be focusing on the nuclease enzymes, the zinc finger nuclease, well-known transcription activator-like effector enzymes, and the CRISPR-Cas9, the focus of our webinar. Then the rationale behind the role of CRISPR system as a component of the bacterial immune system and the response to infection with these tiny particles called the bacteriophages will be placed on the second axis, focusing on the endonuclease and the process by which they acquire their immunization through these infections. We'll be then briefly overviewing the principle, the mechanism, and the major outcome gained from the use of the CRISPR-Cas9 technology in genetic manipulation. A brief summary of the major useful application in the different fields of interest will be also mentioned at the end of this webinar. So the genome editing effort that lead to the rise of functional tools start with this image from the New York Times 18 years ago. On today's date, June 27, 2000, the New York Times announced in the head news the cracking of the human genome code by a group of scientists, launching the race for the complete sequencing of the human genome that has been later published in one of the most prestigious and peer-reviewed journal of Nature Reviews in 2001. So, since its publication, the Human Genome Project has been considered as one of the most exciting breakthroughs in the human genetics in modern time after Mendelian genetics many years ago. Geneticists from around the world collaborated to determine the nucleotide sequence for the complete human genome, providing the genetic map of the spatial distribution of thousands of genes composed of roughly 3 million nucleotides that are distributed all over of our chromosomes. Later on, the personalized sequencing, only recently applied to clinical medicine, has already been broadly applied to the discovery and the study of disease. It's expected to enable the earlier and the more accurate diagnosis disease and occurrence, guide prevention and facilitate the monitoring of health and treat patients, and play a role in the prevention and the recurrence of future disease. As you know, the human genome sequencing has enabled large-scale genomic studies of different populations and has become a powerful tool for studying diseases, prediction, and diagnosis. The sequencing techniques characterize individual genome, transcriptional state, and genetic variation in population, and then the diseases that are related usually to mutations that affect one or more than one nucleotide in the genes that are responsible for the development and the synthesis of proteins involved usually in these diseases. We have to note that the potential for personalized sequencing today is very important in the diagnosis of cancer and pharmacogenomic application to develop the most adaptable and promising proteins and drugs that can target these genes in order to promote the development of protocols that lead to the elimination of these causing agents, eliminate these mutation effects on the cells and later on in individuals. Here we have to note that the scientific progress and the evolving tools, including the process of DNA sequencing, either the chemical or the enzymatic, the genetic cloning, and the gene manipulation by using this recombinant DNA technology, the genome editing, shaped by the molecular and clinical advances, they now enable an holistic and complete analysis of the individual human genomes to allow practical and productive application to research questions, and more recently, the personalized clinical evaluation. So the genomes of eukaryotic organisms, as you know, they are composed of millions and billions of DNA bases. So our ability to change these DNA bases at precisely predetermined locations hold tremendous value, not only for molecular biology, but also for medicine and biotechnology later on. Therefore, introducing these desired changes into the genome, what we call today the genome editing, has been a long sought after goal in molecular biology. To this end, the discovery of restriction enzymes that normally protect the bacterial cells against infection with microorganism of microorganisms, today we coined the name of phages, of bacteriophages, in the late 70s, was a turning point that fueled the era of recombinant DNA technology. For the first time ever, the scientists gained the ability to manipulate the DNA in the test tube, to cleave the DNA, to shape the DNA, to select sequences, and to cut this DNA, to cleave this DNA at a specific site using proteins that are extracted from the bacterial cells and that can be used to trim this DNA and trim other DNA that will be called later on the vectors in order to create what we call the recombinant DNA system that paves the road for the development of what we call the genetically modified organism, the GMOs. Also, such efforts drove a number of discoveries in molecular biology and genetics. The ability to precisely alter the DNA in living eukaryotes came a few decades later. One of the initial breakthroughs, essentially in the field of genome editing, came from the realization that introduction of a double-stranded break at the target site increased the ability of the scientist to increase or to insert DNA within these genome, the integration of the DNA. So many research groups, they focus on developing different strategies to achieve what we call the targeted double-stranded breaks, starting from the discovery and the use of what we call the homologous recombination mediated targeting, to the system, what we call the Clolux system, which is a system composed of an enzyme, the recombinase enzyme, and two sequence of DNA, the LuxP system, that flank the DNA from one side and the other in order to cleave and remove a DNA from a cell. These systems were later on followed by the discovery of what we call the zinc finger nucleases, and later on the well-known transcription-like effector nuclease, the TALEN, that are discovered in Xanthomonas bacteria, precede the breakthrough of this era. The CRISPR-Cas system composed of DNA and an endonuclease enzyme that is called the Cas enzyme, that is today, starting from 2013, could be used and be considered as a system for genome editing. We have to note that in the early studies, the researchers utilized direct cutting endonuclease enzymes that they call the meganucleases. Usually they are composed of many subunits. They are multi-subunit enzymes that can be either extracted from bacteria or they could be genetically designed and engineered in order to cut the DNA. So these endonucleases, they recognize large stretches of DNA from 14 to 40 base pairs, and when they were used, they increased the genome editing efficiency. However, they had two major drawbacks. The first one is that these meganucleases, they have a unique recognition sequence. So we have to have the same recognition sequence at any level of the DNA to be cut, which is not simple to have. Secondly, and more critically here, is that the majority of induced double-stranded break are repaired through what we call the error-prone non-homologous and join process. So we have a set of proteins that can repair the DNA that is not templated after this double-stranded break. And here, the process of non-homology and joining will introduce at this level of the DNA random nucleotide, or it could remove random nucleotide, and this is what inserts DNA or delete the DNA. At the end of the process, the gene will be knocked out or turned off. When the process takes place by what we call the homology-directed repair, the process is completely different. Since if we have a donor template or a donor DNA here, this donor DNA could be inserted at the level of this neck or this double-stranded break. If we have a precise cutting process from one side and the other at the level of this target DNA, the insertion of this foreign DNA at the level could lead to the addition of novel capacity or novel character for the organism that gain this DNA and that can be edited by this manner. Later on, the discovery and the utilization of what we call the eukaryotic zinc finger proteins started a new era in genomic targeting and editing. These zinc finger are what we call the zinc ion regulated small protein motifs. So they bind to the DNA in a sequence-specific manner. They recognize a sequence, and this sequence is a module of 3 base paired DNA sequence. Therefore, unlike these meganucleases, the multiple zinc finger modules could be assembled into a large complex to achieve higher DNA binding specificity, and later on, to increase their specificity, the researchers created programmable nuclease protein by fusing these zinc finger proteins with a DNA cleavage domain of the FOC endonuclease enzyme, which gave or provide these nuclease more specificity in targeting and recognizing the DNA to be cleaved. Now, the zinc finger nuclease generates substantial excitement as a genome engineering tool. And then the discovery of the transcription activator like effector proteins from Xanthomonas bacteria can specifically recognize not 3 base pair, but only one single base pair instead. And that inspired further excitement about these proteins, how they could recognize one single base pair and then induce these cleavage. Usually, like the nuclease or the zinc finger nuclease, they were able to create, or the scientists were able to create, what we call the chimeric fusion of the FOC1 DNA cleavage domain, and that combined these domains to the TAL modules, which serve as effective programmable nuclease called the TAL. All of these components, they are dependent on the presence of one DNA sequence and a protein that recognize this DNA. So we are based on 3 different components that are based on DNA-protein interaction. And every time these proteins have to cut the DNA, they have to have a specific sequence. So in order to design an experiment, every time you have to design a protein that recognizes the DNA. In contrast, our newly discovered system, the CRISPR-Cas9, is an RNA-guided system that have the ability and the advantage of only needing or only requiring the design of an RNA that recognizes a sequence at the level of the target DNA in order to induce the process of cleavage. So here we repeat, in every time we have a double-stranded break, which is the origin or the essential tool for the process of genome editing, 2 possibilities figure in the subsequent steps. Either a process of filling the gap with non-homologous and joining proteins that fill the gap with non-templated nucleotides. So they induce what we call the indels, which are the insertion and the deletion, leading to the knockout of these genes. In contrast, if we have or we introduce a donor DNA, imagine that this DNA will be introduced within a plasmid or a vector. So it could be inserted by what we call the homology-directed repair that requires the presence of 2 homologous sequences, inducing a homologous recombination, leading to the insertion of these donor DNA inside of these gap, leading to the addition of a gene or a functional gene that leads to what we call the knock-in. So knock-in and knock-out could be the result of a process of double-stranded break by these systems. Okay, so here we have to separate between 2 sets of experiments. The first one is the set of experiment based on DNA protein interaction, the meganuclease, the zinc finger nuclease, and the talon. All of them, they require the design of a protein that have to recognize the DNA. In contrast, the CRISPR-Cas9 will be RNA guided essentially, so we need for an RNA. There is no protein engineering that should be required, so its time and cost is better than these techniques, do not need for a long time. They have a high specificity, and even we are looking over a number of experiments that increase the specificity of these CRISPR-Cas9 and is multiplexable. It means that inside of the same cell, you have the ability to introduce a system that is based on the CRISPR-Cas9 in order not only to target one single gene, but it could be used to target and alter many different genes at the same time. And this is what is needed for the treatment or the manipulation of cells, especially the cancer cells that are known to have and to be related not only to a single mutation, but to a large number of mutations that are essential and that are behind this process of disease development. So usually the process here will be focused on a system, and programmable system, an RNA-guided system that will be used to cleave the DNA and then to introduce inside of the cell new genes or functional genes. What is the importance of these CRISPR-Cas9 in the life of the bacteria? These bacteria, as you know, they are prokaryotic cells. They are not eukaryotes. They are not doted with and developed immune system. So they have to develop strategies in order to protect themselves against other microorganisms, like, for example, the bacteriophages and other mobile elements. First of all, as you know all, the viruses, they are common threat for cellular life. Now, the least to bacteria and archaea, who constitute the majority of life on Earth. Consequently, a variety of mechanisms to resist viral infection has evolved in both the prokaryotes and the eukaryotes. Now, the restriction modification and the abortive infection in bacteria are examples of such innate system in prokaryotes. This is not sufficient. These prokaryotes have to develop what we call the adaptive system. The adaptive systems have the ability to learn and to recognize specific features of pathology. I would like just here to mention that in our cells, in a human, the B and the T cells, they can learn to recognize the proteins, the foreign proteins. They learn to recognize the invaders and others. We all remember that we have what we call the macrophages, the antigen-presenting cells, and the effector immune system cells. So anytime we are infected with an invader, this invader will be captured by our immune system cells and will be expressed at the surface of our cells in order to activate the development, the proliferation, and the maturation of both the B lymphocytes and the T lymphocytes. In both of these pathways, the immune reaction will end up by increasing the effectiveness of this immune reaction, and then the development of what we call the memory cells. And this is what happens when we are vaccinated. We maintain in our body what we call the memory cells, and these memory cells will serve for a later infection. If we are infected with the same viruses or bacteria, will be protected, since our memory cells will be present, will be ready to activate the development of an immune reaction that will be heightened and that will be more effective than a primary response that requires a long time, weeks, before it could be able to secrete immunoglobulins or even to activate the cytotoxic effect. What about the bacterial cells? Our bacterial cells, the bacterial cells, they are living organisms. They could be infected with bacteriophages. These bacteriophages, as you see here, they are structures that are composed of proteins that constitute their head, polyhedral head, in which nucleic acid, here represented the DNA, but they could be, or they could enclose inside double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA, whatever of their genetic makeup. These bacteriophages, they have a tail, and they have their fibers that serve to bind the surface of the bacterial cells. When they bind the surface of the bacterial cells, they inject, this is an electron microscopic image of these bacteriophages that bind to the surface of the bacterial cell in order to induce the injection of their genomic DNA inside of these bacteria. So if this DNA will be got by the cytoplasm, this DNA carries the different genes from 4 to 100 of genes. Okay, so they could have genes that can be used and that can use the cellular machinery in order to synthesize novel proteins of their heads, proteins of their fibers, and tail in order to assemble inside of the bacterial cell, leading to its lysis. Okay, so they have what we call the lytic cycle. The lysogenic one, they include their DNA within the bacterial genome in the form of prophage. But those that have lytic activity, and they are the majority, they lead to the lysis of the cell. So the bacterial cell, if they do not develop their system of protection, they will be lysed. Their system are either the specific nucleases that serve as a part of their innate immune system, so they recognize these foreign DNA and use their degradation, or the CRISPR-9 that will be able to recognize these DNA and then to develop a system, to develop the capacity to recognize what is inside of these DNA, what genes are carried by these DNA, what sequences, repetitive sequences, or the fingerprint of these virus that will be recognized by the bacterial cells, and that will represent the antigenic determinant used to develop the memory response or the memory of these stages for a later recognition and destruction. So here, in a schematic way, the bacterial cell could be infected in a primary way with the viral DNA. The CRISPR-Cas9 system is a complex that is composed of what we call the CRISPR loci or locus and a specific catalytic enzyme. We'll be talking in detail about this enzyme, the Cas9. So the viral DNA will be recognized by the bacterial cell protein, and then they will be degraded. Fragment of this DNA here, the fragment of the DNA that will be cleaved by the Cas9 protein or endonuclease, will be used in order to insert this fragment within this locus, and that will be served as a memory for this bacterial cell. If the bacterial cell will be later on infected with the same bacteriophage, here the Cas complex in the presence of the RNase 3 and a transactivating RNA molecule will be used in order to synthesize a specific hybrid system that contain the CRISPR RNA and the transactivating RNA that will be associated with the Cas complex in order to induce the cleavage of these DNA. How? Usually, this structure will be inserted within the gene, so it will become expressed or ready to be expressed by the RNAs. So usually, the RNA polymerase. These RNA polymerase will create an RNA that will guide essentially the Cas complex in order to recognize this DNA of the bacteriophage, leading to its degradation. So usually, the process requires the immunization step at the first encounter with the bacteriophage, and then insertion within these repetitive sequences containing spacers. These spacers, they are intercalated by these colored structures, and every one of these colored structures represents one of the DNA of viruses or bacteria that were recognized previously by these bacteria. So if the bacteria survive an bacteriophage infection, they will add to these sequences one additional memory sequence that will be used to induce the synthesis of these RNA. So these sequences will be separated by repetitive sequences that will be duplicated before they are expressed in the form of CRISPR RNA. This is first observed in E. coli, and it has been demonstrated that it is present in about 84% of archaea and 45% of bacteria. Now, usually the spacers are derived from nucleic acid of viruses and plasmids. Here, these observations give us the idea that this CRISPR could be a part of the antiviral system of the bacterial cell. Here, these animation represent what happens at the level of this DNA that will be associated with the CRISPR-Cas9. So the CRISPR-Cas9 should have an helicase activity that lead to or that allows the separation of these DNA into two single strands, and the attachment or the binding by homology and complementarity of these single guide RNA or these CRISPR RNA that will guide these Cas9 or these endonuclease to the target DNA at the level of the cytoplasm. In general, the CRISPR-Cas9 mediated defense can be divided into three major steps. The first one is the process of adaptation in the normal cell that leads to the insertion of new spacer in the CRISPR locus. So every time a spacer will be added, it represents one of the memory for the bacteriophage that has been attached to the DNA, to the surface of the bacteria that injected their DNA, and that DNA has been acquired. So this is what we call the adaptation. Later on, in the second stage, expression. The stage of expression means the system gets ready for action by expressing the Cas9 genes and transcribing the CRISPR into a long precursor CRISPR RNA that would be subsequently processed into mature CRISPR RNA by the Cas9 enzyme and the Cas9 protein, in addition for sure of other accessory factors. And then in the later phase, what we call the interference. Why interference? Because here the RNA interfere in the process of elimination. What interfere here is the guide RNA, here this structure that have the thread part that recognize the DNA at the level of the complex constituted by the CRISPR for the endonuclease, the Cas9, that possess the capacity to separate the DNA and to bind these complementary. Now, how this CRISPR-Cas9 could be used as an efficient tool for genomic? Here we have to note that for the first time ever, the researcher had an extremely flexible tool that could be easily guided to target the nearly any location in the genome by simply designing a short guide RNA. So here we have a sequence that is considered as the target. The scientists have the ability to target these DNA specifically by an process that is RNA guided. Due to the high efficiency and ease of use, the researcher from diverse fields quickly adopted the CRISPR technology as a method for choice for various genome targeting processes. Look here. They are widely used today. And since its inspection as a genome editing tool in the late 2012, more than 9,000 research articles have been published about it. And the number of publications seems to increase every year. The number of PubMed publications over the last 12 years that had the CRISPR or the Cas9 as key terms in their abstract are demonstrated here. So CRISPR publication, we collected every publication that possesses in their title or in the abstract the CRISPR or the Cas9. Here we are today in the middle of 2018, and the number of publications in 2000 is projected to be more than 5,000 per year. So let's focus on these systems. The CRISPR What means CRISPR? It stands for clustered, regularly interspaced, so they are clustered within the same sequence, within the same region. They are regularly clustered and interspaced by short palindromic repeats. So they later on they were called this acronym CRISPR, and we demonstrated that they are not palindromic. So we are using these long term abbreviated with the CRISPR that has been discovered later on to be non-palindromic. But we have repeated sequences. These repeated sequences, they form an array composed of many repeats. These spacers, okay, or these sequences, they are gained. Every one of these colored structure is gained from a previous viral infection of these bacteria. They are preceded upstream with a promoter that could be used to express these genomic material, and a Cas operon that constitute a sequence of genes, family of genes, that encode for the Cas9 and or other system or other proteins, the Cas1, Cas2, etc., preceded also by a promoter and a transactivating RNA. So this structure, it became very important, and the crucial work based on this system that marks the beginning of the CRISPR as an biotechnological tool has been the demonstration that the Cas9 enzymes, especially the Cas system proteins, can be reprogrammed to target a desired DNA in bacteria. What is important in this system is that these structures or these proteins could be effective in different cells and not only in their original or source protein. Now, here we present the structure of these Cas9 from which we were based to develop the genetic editing. So in the natural bacterial CRISPR-Cas9, short sequences that are called the protospacer. Protospacer are the structure of these DNA before they become the spacers within these locus of the Cas9. So they are located and they are part of the bacteriophage DNA, but not the genomic DNA of the bacterial cell. Every one of these structures will be treated, will be processed before it could be introduced within these locus. It seems that these repetitive sequences of the bacterial chromosome should be duplicated before any one of these spacers will be introduced. So every one of these spacers will be used by the cell, by the RNA polymerase, in order to synthesize what we call the CRISPR RNA that will be used in order to target this DNA once the bacterial cell will be subsequently infected with the same bacteriophage. Here we have to note an important feature of this system, which is, and the question is to be asked, why this system do not affect and do not cleave the bacterial own DNA? Why this DNA is not altered? In the innate immunity, the DNA of the bacterial cell is either methylated or semi-methylated, acetylated, so the restriction enzymes will not be able to alter this genomic DNA. In the case of the CRISPR system, this Cas enzyme is unable to target the bacterial DNA since they require the presence of what we call the PAM. Okay? This PAM is a protospacer adjacent motif, and that is proper for the bacteriophage DNA, but not the bacterial DNA. So the Cas9 do not bind and do not alter a DNA without the presence of the PAM. In the case of some bacteria, okay, and mostly is an NGG. So if they recognize and encounter these PAM, they can then bind and cut this DNA. Otherwise, the bacterial DNA will be completely protected. So this protospacer adjustment motif is an essential part and essential feature, essential requirement for the process of protection. Today, we have a large number of studies that are running in order to find and in order to identify the presence of these PAM sequences. These PAM prevent the cleavage of the bacterial own genome at sequences that are complementary to the CRISPR RNA. But since the simplicity of the Streptococcus pyogenes PAM sequence, it could be considered and used for a large number of applications. Today, we have a large number of studies that identify many PAM sequences, and they are exploring other CRISPR systems to identify the Cas9 effector proteins even that recognize these different PAM sequences and the PAM requirement and even the substrate requirement. This is because they need to alter at a very high specificity. So look at this structure. This is the Cas9. This is the single guide RNA. That is the assembly of the CRISPR RNA, the transactivating RNA, in the same complex in order to form what we call the single guide RNA. The genetic engineering of this system allowed the formation or creation of this single guide RNA, including both of these structures, in the presence of the Cas9 complex, in order to use it as a key term and key component of this genetic editing or genomic editing. So here we have to explain how this single guide RNA could be produced. We have to say that in the Streptococcus pyogenes system, we have two RNA sequences. In other bacterial cells, other system could be considered. But here, we consider these type of bacteria because of their simple PAM sequence. The CRISPR RNA and the transactivating RNA form a hybrid molecule that will direct the Cas endonuclease to the target site. These two molecules, they were engineered to combine into a single guide RNA that is approximately 80 to 100 nucleotides, and that have the form of these stems and loops. These stems, they are formed by complementarity between these nucleotides of the RNA. Every time we have a sequence without complementarity, they form a loop. And the secondary structure of these single guide RNA is the appropriate structure in order to allow the Cas9 to bind it, leaving these CRISPR RNA sequence to recognize and bind the target RNA and bind the target DNA. Here, these sequence recognize the PAM, find the PAM, and then induce the alteration or the break at the level of the DNA. Without these PAM, or the mutation of these PAM would affect the specificity of the CRISPR sequence. Many studies, they are today running in order to what? In order to prevent these off-target. At any time, these Cas systems find this sequence of nucleotide within region outside the target of alteration. We have also to say that the Cas9, in addition to these single guide RNA, when they bind to the DNA, they should induce the separation. So they have to have two different activities that are the helicase activity and or the nuclease activity. Both of them will serve in order to cleave and separate the DNA. Okay. Here also we have to note that the sequencing or the genomic sequencing techniques and the new generation of sequencing allowed us to study further these Cas systems, and we classified them into two different classes according to their original bacteria. So we have two different types of CRISPR system. The class 1, and this class 1 is composed of what we call the multi-subunit CRISPR RNA effector complex, in which this complex is composed of a number of subunits in order to form the Cas endonuclease. The class 2 usually is composed of a similar sized region, but that will compose an unique structure. So they are called single subunit CRISPR RNA effector complex. In this case, they will produce a protein that will be used for the process of genetic or genomic editing. These are characteristics of the type 2. So look at the class 1, the type 1, the type 3, and the type 4. All of them, they have multi-subunit structure of their CRISPR RNA encoding region. In contrast to the class 2, and specifically the type 2, why the type 2? Because this type 2 is preceded by the RNA type 3 that will be used in order to cleave the RNA and create the CRISPR RNA and the transactivating RNA, in the presence of the Cas1 and the Cas2 that will be used to cleave the DNA in order to form the complex or the effector complex. So returning back to this structure, we have the type 1, the type 2, and the type 3 essentially. Look at these type 1, type 2, and type 3. The Cas9, the Cas genes, the locus of the CRISPR, and the promoters. So usually the Cas system will synthesize these CRISPR RNA, but the CRISPR RNA of the type 1 and the type 3 will be multi-complex structure with the PAM proteins. So if they recognize and bind a specific sequence, they require the presence of all these subunits. So the deficiency in one of these subunits will alter the activity of these type of Cas enzymes. In contrast, the Cas9 is usually composed of only one single subunit, the Cas endonuclease, and the PAM structure at the level of the DNA will be recognized by this CRISPR RNA. All of these systems will lead to the degradation of the RNA at the end of the process. So we are focusing on the use of this type 2 for the process of development of these systems. Now, before ending our seminar, we have to summarize the major therapeutic applications of this CRISPR system. And we have to say that today we are not yet at the clinical level. We're still at the research basic level, but we have a large number of promoting or possible applications of this CRISPR, specifically in the human therapeutics and the targeting of genes that are behind the development of diseases, namely the cystic fibrosis, the beta thalassemia, many other diseases that are related to these genes. In agriculture, in order to target genes that are involved in the synthesis of proteins leading to the alteration of the process of flowering, the process of maturation of the plants, or the development of diseases, the drug discovery, and here get into the same part, the specialized or the personalized medicine with the CRISPR system in order to synthesize or to find the most appropriate sequence of the DNA leading to the synthesis of protein responsible for the disease development in order to design a therapeutic protein that targets only and only this altered protein. So here we can use the CRISPR in order to specialize the process of treatment. Also, this CRISPR system could have a very large application in industrial biotechnology, specifically in the targeting of bacteriophages that alter or infect the different machines and pathways in the process of their reproduction, and even the gene drive where we can use these CRISPR techniques in order to change the process by which genes can be transmitted from one generation to the next throughout the process of inheritance. And this will help to disseminate, for example, some genes in population of bacteria, of plant, of any other organism with a specific functional gene. For the clinical trials, we have to wait the FDA approval. This FDA approval requires a large number of requirements before it could give any of the research groups the ability to test their system or their product in the treatment of diseases. Today we have a number of groups that have some preliminary trials, especially the clinical trials type 1 and 2. We have a group in China that started a clinical trial for the treatment of multiple myeloma and the alteration of or the manipulation of lymphocytes that could be modified by this CRISPR system in order to recognize and bind the cancer cells for a process of inducing the apoptosis or the process of apoptosis in these cancer cells. In any one of these applications, there is a major requirement that have to be set at the level of the cell, so in vitro, ex vivo, or even in vivo, we have the same requirement. We need for a 20 nucleotide target sequence that should be inserted within a vector. The vector will be used to synthesize the single guide RNA in order to bind the target sequence. So the single guide RNA will contain the CRISPR RNA that recognizes the target sequence. They should be preceded by a strong promoter, and this system will be introduced in the cell containing an antibiotic-resistant gene and origin of replication in order to recognize the cells that acquired this system and start the process of propagation. This system is functional. Inside of these cells, they will be able to synthesize two different types of protein. Here we have a vector that has been designed to synthesize a single guide RNA that will recognize and bind the target sequence, and the Cas9 gene that encodes for the Cas endonuclease and origin of replication, and a beta-lactamase encoding gene. So this gene will be used to recognize the cells that acquire the vector. So usually the single guide RNA and the Cas9 coding sequence can be expressed in these cells. When the genes are introduced, the promoters that drive the expression and the endonuclease, they bind the transcription factor, and then we have to optimize this process of synthesis by using the most optimized structure of their promoter. The donor DNA usually is flanked by sequences that are homologous to the target genomic site for insertion by homologous recombination. So usually the vector is introduced into the recipient cell, and following the expression of the single guide RNA and the endonuclease, usually the recipient cell genome is cleaved at the target site, and the activation of recombinase that mediates the DNA repair will result in the recombination process that will lead to the insertion of the donor DNA in the genome at the target site. So if we require, or if the experiment requires the presence of a specific gene, this specific gene will be also present. Now, one of the experiments that we're running at a preliminary level, we have the one that used the gene editing therapy for the CTX001. What is that? Usually the blood disorder beta thalassemia could be the first human disease to be treated using the CRISPR-Cas9 technology in Europe. So we have the process with the ethical committees that are studying the whole process and the whole protocol. The team, or the experimental process launched by the team, they submitted the application for the phase 1 and 2, I think, in Europe to test the gene editing therapy for the first time, and the trial is expected to begin at the end of 2018, as I read. We have another second application for the tire in the US to test this CTX001 in another blood disorder, which is the sickle cell disease. So we have the beta thalassemia and the sickle cell disease, which is also planned for the end of 2018 and the start or the first trimester of 2019. Usually, this CTX1 is what is known as an in ex vivo CRISPR therapy. What means that? It means that the gene editing is done outside the patient body. Usually, the hematopoietic stem cells are harvested from the patient. They will be engineered in the lab to produce the red blood cells with high level of fetal hemoglobin. And then the newborn naturally produces this protein that can bind the oxygen much better than those of the adult with altered hemoglobin. Now we have to say that the engineered cells are then transfused back to the patient where they can treat the genetic disorder that impair oxygen transport in the blood, such as the beta thalassemia and the sickle cell disease. As we said, we have a Chinese group that is working on this PDL or the PD1 and the PD1L, which is the ligand and the receptor. Usually the tumor cell can inhibit body's immune system by binding to the protein at the surface of the cell, which is the PD1 on the surface of the T cell. So the T cells will become ineffective. What is the importance of the CRISPR-Cas9 is that they can turn off the PD1 gene in order to reactivate the immune system. So what happens here is that the cells are extravasated, they are cultivated in vitro, and then to achieve the goal of altering, so the gene coding for the PD1 protein, which is the program -1 protein, is switched off by using the CRISPR-9 technology. And then when they are returned back to the body, these cells or these modified lymphocytes will have a more effective capacity to bind these cancer cells, the multiple myeloma cells, and induce their cell death by apoptosis. So the hope here is that these modified lymphocytes will be able to alter their level of PD1 and to induce the signal at the level of these multiple myeloma cells that will proceed in the process of cell death by apoptosis. Another group also has been working on what we call the Cas9 mouse. They are transgenic mice that were transfected at their embryonic level by the Cas9 gene associated with an GFP protein in order to identify the tissues that are able to express this protein, preceded by a strong promoter and a specific sequence that lead the Cas9 to bind their specific sequence. To end this talk, an overview about the different field or expected field of application for this CRISPR-9 system, the gene editing as we discussed during our talk, the gene regulation, the epigenome editing by binding a specific modifier or transfector enzyme that alters the structure of the DNA and their capacity to induce the expression of the gene, the chromatin imaging and their distribution, the special distribution inside of the nucleus, the base editing specifically at the level of specific proteins for like the ApoB involved in the regulation of the cholesterol level in the blood, the RNA targeting, especially those that are involved in the process of interference and the alteration of the genetic level of regulation, the chromatin topology within the spatial and the temporal distribution of these chromatin within the nucleus, and that is related to the presence of transcription factors involved in their level of expression and the chromatin imaging. I would like to thank every one of you for your attention and will be waiting for your questions, if any, about this presentation.